Hello everyone, I'm Sangeeta Dwivedi, Assistant Professor of Modern Institute of Pharmaceutical Sciences, Indore. Today I'm going to deliver my lecture on bacterial growth curve. So typically a bacteria and archaea reproduce by asexually mode, and while eukaryotic microbes can engage in either sexual or asexual reproduction. So bacteria most commonly engage in the process known as a binary fission, where a single cell splits into two equally sized cells. Other less common process through which a bacteria can reproduce are a multiple fission, budding, and the production of spores. So bacteria require certain condition for growth, and these conditions are not necessarily are same for all bacteria. So after reproducing through binary fission, the mass and number of bacteria can increase in exponential manner, and uh, the bacterial growth phase we can divide it into four distinct phases. And here I can also included a point that is showing generation time. This is the time which is required for a bacterial cell to divide, and the population, or we can say the time of microbe to get double in their number. Like here, for every bacterial species it is specific. Like in the case of Escherichia coli, the generation time is 20 minutes, means in the 20-minute Escherichia coli get doubled in their number. So after 7 hours, a total 20 generations may produce of Escherichia coli. So one cell becomes 1 million cells after 7 hours if we are talking about the Escherichia coli. Here the picture shows the binary fission mode of reproduction of bacteria. This process, binary fission process, begin with the cell elongation, which requires a careful enlargement of the cell membrane and cell wall. In addition to an increase in the cell volume, the cell starts to replicate its DNA in preparation to having two copies of DNA in each newly formed cell. So this picture is clearly shown that how number would increase after each generation. So this is showing the number of cells, and so here the number of bacterial cell is 1. After one generation, the number of bacterial cell is 2. After 2 generation, the number of bacterial cell is 4. After 3 generation, the number of bacterial cell is 8. After 4, the number of bacterial cell is 16. And after 5th generation, the number of bacterial cell is 32. So since bacteria are easy to grow in the lab and their growth has been studied extensively, it has been determined that in a closed system or batch culture. So what does it mean? Batch culture means when no food added and no waste removal is performed. So in the closed system or batch system, bacteria will grow in a predictable pattern, and it resulting in a growth curve which is composed of 4 distinct phases. One is lag phase, second is exponential or log phase, third is stationary phase, and the fourth and last phase is death or decline phase. Additionally, the growth curve can yield generation time for a particular organism, and as I can previously explain that the generation time is the amount of time which takes a population to get double. So the first phase is lag phase, and this is an adaptation period where the bacteria are adjusting to their new condition. The length of the lag phase can vary accordingly based on how different the conditions are from the conditions that the bacteria came from, as well as the condition of the bacterial cell themselves. Typically, cells in the lag period are synthesizing RNA, enzymes, and essential metabolites that might be missing from their new environment, such as if we are saying that like growth factor or many macromolecules. as well as adjusting to environmental changes like temperature, pH and oxygen availability. They can also be undertaking any necessary repair of their injured part. So here in this phase, basically bacteria can increase in their sizes, increase their metabolic rate, and they are trying to adopt a new environment and necessary enzymes. The next phase is logarithmic phase, or also known as exponential phase. So in this, once the cell have accumulated all that they need for their growth, they proceed into their cell division. So exponential or log phase of growth is marked by predictable doubling of the population, where one cell become two cell, a two cell become four cell, as I earlier described in the picture in slide 3, and four cell become 8, etc. So conditions that are optimal for the cells will result in very rapid growth. And in the curve that we will see in later slides, the rapid growth will be shown by a steeper slope on the growth curve, while less than ideal condition will result in slower growth. So cells in the exponential phase of growth are the healthiest and most uniform phase of cells, which explain why most experiments utilize cells from this phase. In most experiments of microbiology lab and biotechnology lab uses this phase cells. Means they use the cells, they prefer the cells when they are in their exponential phase, because they are in their healthiest phase in exponential phase. And the third phase is stationary phase, and stationary phase comes after when a rate of multiplication and death rate become almost equal. So uh, at some point the bacterial population runs out of an essential nutrients or their enzymes, their growth factor, uh, or its growth is inhibited by its own waste products or lack of physical space, causing the cells to enter into stationary phase, where the multiplication and the death rate are become equal. At this point, the number of new cell being produced is equal to the number of cell dying off or growth has entirely ceased are same. The population growth is limited by three factors. One is exhaustion of nutrients, second is accumulation of waste products, and third is lacking of space, which is known as lacking of biological space. The fourth phase is decline phase or death phase. In the last phase of growth curve, the death phase or decline phase, is in this phase number of viable cell decreases in a predictable or in exponential fashion. The steepness of the slope in their growth curve corresponds that how fast cells are losing their viability, how fast cells are dying. So this has happened because they are getting nutritional exhaustion, toxic accumulation, and their self autolysis of enzymes. So this is the picture which is self indicating the various phases of bacterial growth curve. This is the inoculum, which containing culture media and few cells of bacteria. So here you can see this is a lag phase. The time period of lag phase is small, and the steepness of curve is also very small, because here the few bacterial cells are adopting their environment to for the readiness of their division. Now this is the phase of exponential growth. Here you can say these are live cells, and uh, they are dividing very fast. The steepness of the curve is very high. This is showing that the division of cells are in their highest rate. The third phase is stationary phase. Here you can see the steepness of curve is lost, means the death rate and the production rate of bacteria are the same. And here you can see in the picture that the pink color is also added, means these dead cells are also coming in the picture while we are talking about the stationary phase. So the production rate of new bacterial cell and the death rate of bacterial cell are equal. Now the last phase is death phase. Here you can see the dead cells numbers are continuously increasing, and the production of division of new cells, production of new cells are continuously slowing down. So thank you so much. This is all about the bacterial growth curve. Thank you.